in the nitrogen cycle, the second step is nitrogen assimilation. So in the first step, the nitrogen fixation is done either by using the physical process or a biological process. In the biological process also, either it may be by asymbiotic or symbiotic manner. Ultimately they are forming ammonia. So here the so formed ammonia will be absorbed by the plants from the soil by using the roots, and once this nitrogen or ammonia enters into nitrogen enters into the plant in the form of ammonia. So here that will be utilized in the formation of various types of organic molecules, like it may be in the form of formation of proteins, amino acids, etc. will be formed. So here the utilization of this nitrogen in the form of ammonia in the formation of various types of organic molecules is, this type of nitrogen is called as a organic nitrogen. The nitrogen which is present in the biomolecules like proteins and amino acids, we call this as a organic nitrogen, and the process of conversion of the ammonia into this organic nitrogen is called as nitrogen assimilation. So this is the way how this the nitrogen will be converted into inorganic nitrogen, or ammonia will be converted into the organic nitrogen, and once the plants are being eaten by the animals. So here once again the bonds will be reformed, and some bonds are broken and some bonds will be reformed, that lead to formation of once again the biomolecules inside the animals also. So that is also one of the step of the nitrogen assimilation, where the nitrogen in the form of organic nitrogen will be formed inside the plants, in the plants and also in the animals, either it may be in the form of proteins or in the amino acids like examples. In the nitrogen cycle, the third step is the ammonification. So this is a process where when a plant or animal dies, so actually the plant and animal which contain the organic nitrogen inside them, that is in the form of proteins or amino acids, so this organic nitrogen will be converted into ammonia. So here the formation of ammonia from organic nitrogen by the decay of death and decay of the plants and animals is called as a ammonification process. So this can be done by some of the species of bacillus bacteria. So the process of conversion of organic nitrogen present in dead and decay organic materials into ammonia is called as ammonification, which is done by some species of bacillus bacteria. Now whatever the ammonia that have been formed, so what will be the fate of ammonia inside the plant? In the physiological pH, the so formed ammonia will be converted into ammonium. In some of the plants, so it may exist in both the ammonia and also in the ammonium form. When compared to ammonia, ammonium is more toxic to the plants. So here how the plants will get rid of this ammonium from the plant, or how this will be reacting with other compounds to form into take off this ammonia from the plant body. So here the first one is a reductive amination. In the reductive amination, here the ammonium will react with alpha ketoglutaric acid. And it will be converted into glutamate. For this process they require some energy also, that is NADPH will be utilized for this process, and the enzyme which is concerned with this process is a glutamate dehydrogenase enzyme. In this process the water is formed and NADP is also been formed. So this is a way how this the ammonia will be utilized in the reductive amination process. That is a way how this ammonia toxicity will be reduced. In the fate of ammonia. The other second step is the transamination. So in the transamination process, the transfer of amino group from to the keto group of the keto molecule. So that is the transfer of amino group from an amino acid to the keto group of a keto acid. So here this can be shown in the form of a reaction that the glutamic acid, so which contains amino group, the glutamic acid which have this amino group, which can be shown in this like this. So which is an amino donor. So we will react with a compound which is an amino acceptor, which is an keto one. So this is a keto functional group. So which is acting as an amino acceptor. So leading to the formation of so this is a way how this the amino donor is giving this amino group to a keto group of keto acid, where so here the keto is replaced by the amino group here. So this is a way how this the fate of ammonia will be there in the transamination process. In this one there are two types of amide groups are there. One is called as aspartate and other one is glutamate. So these two are the amides. So these are the main structural parts of proteins. And these two amides are the part of two amino acids, that is aspartate is formed from aspartic acid and glutamate is formed from glutamic acid. So these are aspartate is formed from aspartic acid, glutamate is formed from glutamic acid by replacing hydroxyl group by the amino group. And here these amides contain more amount of nitrogen when compared to the amino acids. That's why they are transported through the xylem tissue. And in some plants like the soya bean, here the nodules of the soya bean will transport the nitrogen in the form of urids. And here generally they have more amount of nitrogen when compared to the carbon molecules. So this is a high concentration of nitrogen to carbon ratio. So this is about the how the fate of the ammonia will be there in different types of plants here.